The big picture here, let's summarize this a little bit, and then we'll continue on. So basically, we had the donation of electrons to the electron transport chain, and we got those electrons from glycolysis, from the linking step, from the TCA cycle, or the Krebs cycle, right? So these are the electrons that we are bringing in on our two carrier molecules. Remember, NAD is a carrier molecule, and so is FAD. Now, the final accepting of electrons must take place by oxygen, right? This is why oxygen is breathed in, and I keep saying that because it's really fascinating. The whole sole purpose of breathing oxygen in is to accept some electrons to make some ATP. This is why if you don't have oxygen coming in, that whole chain of reaction halts, and you basically stop making ATP, and you basically die. This is why you need oxygen, right? Okay. So we had the oxygen accepting those final electrons and then making water, essentially, two H's, and that oxygen makes H2O, and that can be released into the body. And then we also want to remind ourselves that we have, along the way, through these processes, we have the release of CO2, which is another metabolic byproduct of this process. Now, the big picture here, we yield 28 ATP, which is much more efficient than the 4 ATP we yielded through glycolysis, the linking step, and the Krebs cycle, right? Those three processes only yielded us 4 net ATP, whereas this final process, oxidative phosphorylation, which requires oxygen, yields us much, much more ATP, okay? Okay, and so let's just walk through this one more time, again, for repetition's sake, just to make sure we understand this. So again, we have our 3 important complexes and then this final ATP synthase. We also have some coenzymes that are in between here. Don't get too worried about those, but these are also involved in that passing of the electrons. So we have our electrons, which are coming from our carrier molecules. So these have been oxidized back to their oxidized state, and these electrons are now liberated to enter this chain. They enter complex 1, they're passed to coenzyme Q, they're passed to complex 3, they're passed, oops, I should be showing this here. Okay, so they entered here, and they go from complex to complex to complex, and then they come out on this other end, right, our electrons. They're gonna be donated to oxygen, and we're gonna yield water here, okay? Now, all the while we're doing this, every time we move an electron across a complex, we're basically pumping some protons across the inner mitochondrial membrane, such that we have a much higher concentration in the intermembrane space than we do in the mitochondrial matrix. And that's what eventually gives us this driving force here. So this is going down its gradient, hence the green arrow, as opposed to going against its gradient, hence the red arrow. Okay, so now we can synthesize ATP much more efficiently this way. Okay, let's look at a review of this entire process. So again, we had glycolysis here, this linking step, the Krebs cycle, and then finally oxidative phosphorylation. Let's look at some of our carrier molecules. This is very important. So you're gonna have to know the sources of our carrier molecules here. Like 2 NADH here, 2 NADH here, 6 NADH here, right? These all enter oxidative phosphorylation, and we eventually yield 28 ATP. So this is a slide you probably want to highlight, make some notes on. This is probably where you want to focus your studying, right? The big picture of this process, where we're getting our carriers, where we're picking up our electrons. How many electrons do we pick up from glycolysis? Where do these electrons eventually go? In terms of oxidative phosphorylation, how many ATP do we eventually yield? How many ATP did we yield before oxidative phosphorylation? Okay? All right. Okay, so this is the overall reaction here. Again, 1 molecule of glucose, let's keep that in mind. And I also want to remind us once again that other substrates can undergo cellular respiration. This is just an example for today. So glucose is the substrate we're talking about. And we eventually yield. Now there's 32 to 38 written on this slide, and that's because there is some variation. Not only is there variation across one text to another, but there's variation within a single individual. So if you're younger, maybe teenage or young adults, you may have a better, more efficient oxidative phosphorylation. So you may yield more ATP. Whereas as you get older, you eventually lose the efficiency of this process and you may start to yield closer to 30 or 32 ATPs. So this can change throughout the lifespan of an individual. It can also change